Hello everybody and welcome to today's webinar. My name is Anna Swan. I'm a training officer here at the EBI. Today we have Anna Hersey from Kemble who's going to be talking to us about the new interface.
Hi everybody. What I was wanting to do today is obviously, as Anna said, focus on the new interface. But I thought before I did that, what I would do, because I obviously don't know exactly what people know about the ChEMBL data, is just show you a few slides that sort of explains the data that we have in ChEMBL, and then we'll go on to working our way through some specific examples using the interface. So ChEMBL is basically a database of bioactivity data. So we've got getting on for 2 million compounds in the database, and primarily the data comes from the medicinal chemistry literature. So we're manually extracting data from the literature, putting it into the database. But in addition to that, we also have data sets that various people have deposited, particularly around neglected diseases, but also data from some pharma companies that has made its way into ChEMBL. We extract data from a subset of data from the PubChem database, and we're also taking some data from patents. So some patents have actual bioactivity data in, and we're putting that data into the database. And then because we consider ChEMBL to be a sort of a resource for people who are interested in drug discovery, we're trying to sort of follow through the drug discovery process and trying to annotate information on compounds where they've actually got either to the clinic or a long way down the development pipeline. So this is just some statistics on the amount of data that's in ChEMBL 26. So ChEMBL 26 was released last week. We've got data, as I say, on nearly 2 million compounds, nearly 16 million activities. And I hope everyone's aware that all the data in ChEMBL is freely available for anybody to use. So, you know, you can use it freely in your own particular areas of research. So this is basically the process that we use. So we have a document. Our extractors basically then read those documents. They extract information about the compounds. We then do a lot of standardization, put that information, including the chemical structures, into the database. We then extract a limited amount of information about the assays. We then map the assays to targets and then register the targets, in particular if they're protein targets, against UniProt IDs, etc. Put that into the database. And then the sort of the thing that links the two together is obviously the activities. You know, that a compound was measured with a particular IC50 in a particular assay against a particular target, and that is ultimately, I think, what people are sort of working their way towards using. So I guess a lot of what people use from the ChEMBL database is information about specific protein targets. So obviously we have a lot of SAR data on this sort of thing, but it's perhaps worth mentioning that we extract all the data from a particular article, including, for example, PhysChem data, sort of ADME type data, pharmacokinetics type data, and also if there's in vivo potency data or any data measured in phenotypic screens, that data will also end up in the ChEMBL database for you to search. Just also mention the concept that we have of targets. So where a compound is binding to a single protein, I guess that's a sort of fairly obvious thing to sort of measure and understand, but we also have concepts of protein complexes. So for example, like GABA-A, which is made up of a number of different subunits. And then also the idea of protein families. So particularly in a lot of older papers, they would just say that this particular experiment was measured against muscarinic receptors, but doesn't mention whether it's M1, M2, M3. So we can't say specifically the actual protein that it was measured against. So we record that against a protein family. And then the sort of non-protein targets, we have data measured in, you know, particular cells, tissues, organisms, etc. And those will effectively be recorded as targets as well. So I thought I'd put this slide up because I think this is important when we come to looking at the data in the database. Is, if you like, we have this sort of concept of Kemble entities, and you'll see when you do searches on the interface, as you can see on this slide, there'll be, you know, you'll do a search and it'll tell you there's so many compounds, targets, assays, documents, cells, etc. So compounds and targets, I think, are, you know, probably fairly self-explanatory. The assays is obviously the specific experiment where the particular bit of data has been measured. The documents are usually the scientific journal that the data comes from. And then if information's been measured in cells and tissues, we've also tried to sort of aggregate that information and mapped that to different ontologies so that you can actually find all the information that's been measured in particular cells or tissues. So this is what the data ultimately looks like. So we started off with those journal articles, and then ultimately what we're giving you is, effectively, you could call it a spreadsheet that gives you information about the compounds, the activities of the compounds, and then that's related to the assays, the targets, and then you can obviously get information and links back to the original source data if you want to explore anything a bit further. I've just put this thing about Kemble at the bottom as a sort of a reminder, if you like, to me to mention that there's a lot of data in Kemble, particularly against the protein targets, that's sort of IC50 Ki type data. And we do quite a lot of work to try and standardize all this information, to standardize the units, you know, whether it's nanomolar, micromolar, etc., etc. And we've also, in order to try and facilitate people wanting to look at the data, we've developed this concept of a Kemble value, which is just basically the equivalent to a PIC50 or a PKI. So if you wanted to look at a whole series of compounds against a particular target, you might find that sometimes the data have been Ki data, sometimes IC50 data, sometimes PKI, etc., etc. So by using the Kemble value, with the caveat that IC50 is not entirely equivalent to Ki, you know, this will give you the sort of, I think, a good indication of whether particular compounds are potent or not against a particular target. And then lastly, I just wanted to mention that we do a lot of work to try and annotate drugs. And what we're trying to do here is taking not only marketed drugs, but also drugs in clinical development, and we try and manually annotate what we think is the mechanism of action. So this is different from this compound's being measured against this particular target and it has a PIC50 of whatever. This is just saying we believe that the reason that this drug cures a particular disease is because it acts at a certain target. So this data is available, and I'll show you where it is later. And then also for drugs and clinical candidates, we've done quite a lot to identify the indications for drugs, whether it's from sort of the marketed information for like Daily Med or drugs at FDA, or for the clinical compounds in clinicaltrials.gov. So that was really what I wanted to just start with summarising. what I want to do now is basically try and walk you through some specific examples of things that I think people working in drug discovery might want to do with the ChEMBL data, and in particular using the new interface, which for people who use the old interface, I hope you'll see that there's a lot of new things that you can do that you couldn't do very well on the old interface. So if I now switch to, so if you search for ChEMBL in Google, you'll get this is the sort of landing page now. I should also say that they're moving some of our servers around at the moment, so I'm hoping we're not going to have any problems this afternoon, but because of this, we have had a bit of disruption over the last couple of weeks, so fingers crossed that we'll be okay this afternoon. So, as I say, this is the sort of the landing page. I'll hopefully take you through all sorts of bits and pieces, but I think what we've tried to do here is make this a sort of equivalent of a Google type search box where you can just type in, for example, the name of a target. So I've just chosen this CCR5 target, and you can immediately see it comes back with suggestions on, you know, what information you might find that sort of matches that search criteria. So I know that this is a target, so I want to have a look at what target information is there against CCR5. And you come back with a page like this, which shows 8 different CCR5 targets that match that search. Now, if you look here, you can see that we have, as well as what I guess most people would expect, the data against the human target. We've got information about sort of mouse, monkeys, rats, mice, etc. And on these lines, you can see information about the number of compounds that have been measured against the target, the number of activities, and then a bit of information about how this breaks down. So you can see that, for the example, for this single protein, most of the data is IC50 Ki data. And then we've got sort of hyperlinks to a target report card. And if you wanted to go and look at the information about the protein sequence in UniProt, you can click on that link and go to the UniProt page. So supposing we're interested in this CCR human target, we can click here, go to the target report card, and this is where we aggregate all the information about the specific target. And you can scroll down the page and you can see there's a lot of different sections of information. If you know what you want specifically, you can also use these links here on the right hand side to go directly to the area that you're interested in. So I guess what I wanted to show you here was perhaps the information that we've collected on drugs and clinical candidates. So, you know, we're just summarizing this information here, in particular looking at the phases. On all the tables that we have in the ChEMBL interface, you can click on these arrows and basically sort the data either from high to low or low to high. And so you can see here we've got information about a specific compound that's actually marketed, and its mode of action is through CCR5, and we've got links to where that knowledge comes from. And then if you wanted to, say, go to find out more information about that particular compound, you can go to the compound report card, and in the same way as we did for the target report card, you've got the links on the right hand side to sort of scroll down through the information, and you can see here the information that we've got about the compound, particularly around the indication. So you can see basically that this particular compound is marketed for HIV, and you've got the links where that information comes from. Then also sort of looking further down the page, you've got links on this information to the bioactivity data on that particular compound. So if I go back, you can use the arrows now to go back to the previous page that you were looking at. And if I scroll right down to the bottom of the page, you can also see links to UniProt, various other protein resources, and perhaps importantly links to PDB, where you've got crystal structures of the target in case you wanted to do any sort of docking of compounds. So that's sort of a very simple example of a target search. We start with another example. So here again, it's another target search. I've chosen this cannabinoid receptor. In the same way, we can search for the target. And then again, if you want to then go and view all this bioactivity data, say on this human CB2 target, there are a number of ways you can actually do it. You can either click that number there, you can click the individual data if you just wanted the KI-IC50 data, or you can use this browse activities, which will then open up all the data. If you are just specifically interested in the human data, say, you can click there and then click browse activities. So for this example, let's just look at the IC50 data, for example. So this is now bringing up a table of all the bioactivity data that's been measured on compounds against this cannabinoid receptor CB2. And we can do a number of things here. We can download all the data by clicking on a link here, and it will download the data, and you can click here and then open it up in Excel or something if you wanted to analyze it in your, in your own way. It's worth saying that we have a sort of a default set of columns that you can actually see in this table, but there are a number of other columns of information that you can choose to view or not view if you so choose. And then I think one of the things that's new in the new interface is the ability to filter the data. So there are some examples, so you can filter them, all the rule of 5 compliant compounds. You can filter on just the most potent compounds, for example. And of course, you can sort the data. So I guess the logical thing to do here is to perhaps have a look at the most potent molecules against this target. So I can, just by way of example, select perhaps the 4 most potent molecules against this target. I can look at the compounds, so you can browse the compounds, see the compound structures. And then one of the things I wanted to show you here was this heat map, which you have to get into through this compound page. So if you click on that, so what we're seeing now is excuse me, information about the CB2 receptor and these 4 compounds that we selected, but also information about the other targets that it binds to. So I chose this example specifically because we've got data on this CB1 receptor. So these heat maps, I think, are potentially useful if you want to look at the selectivity of particular compounds. So from this plot, if you hover over them, you get information. You've got the structures, and then if you hover over the target information, you've got information about the targets. If you click on the individual squares, you can actually see a summary of the information of what's been measured against that target, and then you can actually open up the activity data. So the data is actually coloured according to the Kemble values, with the dark values being the most potent molecules. So this enables you to see, I think, that, for example, that compound there, although it's very potent, is not very selective, whereas maybe a molecule like this is much more selective for CB2 over CB1. So that's a number of examples of searches starting with a particular target. The other thing I think people want to do in ChEMBL is to look at compounds from a compound perspective. So if you wanted to just draw a structure that you were interested in, you can open this draw a structure box, and then you've got a sort of a typical structure drawing box that you can draw the structure that you're interested in. But the other thing that I think is worth pointing out is if you've got a molecule, so you can type in the name of a drug, for example. So I've got this rimonabant compound, so I can go to the compound report card, and at the top of the page where you've got the structure, if you click on this structure search box, it then takes you, it puts that molecule into the structure search box, so you don't need to draw it yourself. You can then immediately just do a similarity search, for example, so you can change the level of similarity that you want to search for, click that box, and in the way, I guess, that you would expect, you've effectively done a similarity search on the compound. You can at any point then go back and edit your search, and it brings it back into this search box. So if instead of doing a similarity search, you want to do a substructure search, you can edit your molecule, do a substructure search, and this has returned a large number of molecules. And you can see what we've done here is we've highlighted the substructure that formed the basis of the substructure search that we were doing. If you don't want to see that, you can use this highlight on and off button here. And if you want to see more compounds on a page, you can, just increase or decrease the number that you're actually looking at. And then, of course, on this page, you can then filter. So we've got a large number of molecules here, 1,500 compounds. So probably this is too many to look at, but you might want to just look at all the molecules that are Lipinski compliant. So you can click on the filters, and you've limited it now to a smaller number of molecules. I think the other thing that perhaps I should mention is if, for example, you wanted to see all the molecules that had a molecular weight between 400 and 500, you do need to actually click on each of these. I mean, it's not perfect, but I guess it's one of the things we'll do in the future. So having got a subset of molecules, you can browse the activities. So again, you're back to this page that's now got all the information about the compounds that contain that substructure, regardless of what targets have been measured, the data's been measured against, in this sort of data page. One thing I'd like to mention is this compound key field. So this data comes from the scientific literature. You can click here and see the paper that the information comes from, and you can actually go to the, a link to the journal article if you want to. But one of the things we do is include how that compound is referred to in the paper. So this compound in this particular paper, or this bit of data on this compound in this paper is referred to, I would imagine, in a table as 9D. So this enables you to go directly to the information in the paper on that compound. A bit more information about this, the compound report card. So this is going back now to the drug compound that we mentioned. We've got all the sort of normal representations of the molecules. We've got the data sources. We aggregate all the data on the parent structure. So it's possible that some of the data is measured on the salt and some is measured on the parent. And we aggregate all that, although we do, against each individual measurement, still record whether the data was measured on the salt or the parent. You can see information here. This particular molecule has actually been withdrawn from the market, so the information is there. You've got information about the mechanism that we believe was responsible for its indication. There's information again about the indications for the compound that I showed you earlier. Related compounds, you can get to the bioactivity data either by the individual data types or all the data on the compounds. And there's some calculated properties on the molecule, structural alerts, and then down here we're sort of aware that obviously ChEMBL isn't the only data resource that's going to have information on the compound. So using our UniChem resource, we provide links to where there is other information on this particular compound, whether it's clinical trials or the links database, a PDB, etc., etc. So you can explore that information yourselves. Okay, so we've covered some information about where there's data in ChEMBL on targets and proteins, sorry, targets and also compounds. But supposing you have a target that you're interested in, like let's say IRAC2, and you look at this and you find, well, we've got one data point in ChEMBL, so potentially if you're trying to understand what binds to that particular target, that's maybe not enough data for it to be useful. So the other thing we can do is to do a sequence similarity search by effectively doing a blast search against this protein. So we go to UniProt. You can identify, say, the human protein. You can get the sequence in a FASTA format, and then all you need to do is copy that, go back to ChEMBL, and then where it says enter a sequence, you can paste that sequence in here and then search against ChEMBL. And so what this is doing is actually looking at targets in ChEMBL that are similar to the target that we've put in. So this takes a little bit of time to run, but hopefully not too long. Let's try once more. But basically what it should do is return a list of compounds back into ChEMBL with the most similar targets shown. Yeah, I don't know why, but this isn't loading back at the moment, but trust me, normally you get a table back that it sorts the data in terms of the similarity to the protein sequence, and then you've got a page that looks a bit like the page where you've got lots of different targets, and then you can you can just select the most similar targets. So in this particular case, I know because I've done this before, the IRAC4 and IRAC1 targets are the most similar to the IRAC2 that we looked at. And although we saw that for IRAC2 there was only one data point, you can see there's a lot of data on this compound that's got reasonable similarity, sequence similarity to the target we're interested in. So you can obviously explore the data, and then you can see that you've got lots of protein structures, for example, on that particular compound. So the very last thing I wanted to show you is some of the other ways that you can get at the data. So it's probably worth mentioning at any point, if you decide you want to go back to the home page, you just need to click on this link here. You can use these bubbles to basically get a page up of all the data. So if I just, for example, this mechanisms goes to the drug mechanisms page. So this is now basically a table of all the drugs that we've got information about the mechanisms of. And there's actually a sort of a text box here, so you can type in a target like GABA-A and do a search, and then you can pull back all the drugs that we think work through the GABA-A receptor. And then there's an equivalent page for indications, etc. And then the other thing is that, well, there's these bubbles, but then we also have other ways of exploring the data. So we have this protein target tree. So if you know that you're specifically interested in, say, tyrosine kinases, you can click here and either open all the information on the protein kinases at different sorts of levels, or you can go to something more specific. This will open the target page with all the information about those targets, and you can then explore the data. And by clicking back on this, it will then expand this back out again. So as well as looking at the just the protein target tree, there's an organism tree. So if you wanted everything, gram-positive bacteria, you can do the same thing. And if you want to look at sort of the gene ontology around different targets, again, you can look at that information. And then the very last thing I wanted to show you is some work that we did last year that we actually published. Is, as I mentioned at the start, there's a lot of information on in vivo data in ChEMBL, and obviously that data is much more complex than IC50 values against targets. So what we've done is we've tried to aggregate some of this information together by annotating sort of similar assays and then putting it together in a sort of a tree. So if you're interested in nervous system and anti-epileptic drugs, for example, you can search this, and then you can go to an assays table that gives you all the assays that have been measured for effectively in assays that we believe are related to epilepsy. So I think that's really where I wanted to stop. I've hopefully shown you a number of different ways of accessing the data. If I just go back to these slides, I can just mention that there are a number of ChEMBL papers that we've published on the data. We have a mailing list, so if you want to subscribe to that, you're very welcome to go to this link. If you have any problems when using any of the data or the interface, you're very welcome to contact our ChEMBL help. We have a blog where hopefully we tell you some of our news and new things we've been doing. And then we put up a ChEMBL quick tour on the EBI training online page. So I hope that was useful to people. So I'll hand back to Anna now.
Yeah, so we do have a few questions already, but if you have further questions, do pop them into the chat box and we can have a look through them.
Why are there multiple columns for the CV2 in the heat map? So that is basically because some of those targets weren't human targets. So the two I was showing you on the left-hand side of the heat map were the 2 human targets. But I think if you hover over the targets further along the list, you'll see that I can't remember. I think one was mouse or rat, but that's basically what they are. The binding site information, can we query targets? There is binding site information in the database. I don't think that you can do that on the interface. So yeah, there is information in the database. I don't think you can get to the information about the I've never looked myself. I don't know that you can get to the residue information. Yeah, I mean the drug annotation, we're only annotating against the individual proteins, not. I don't think you can see the actual binding sites. Yeah. Oh yes, so I wrote the stars as just like a wild card. Yes, so you know that was going to give you GABA, you know, GABA A, GABA anything else. Yeah. How can I retrieve toxic compounds? So we do have an assay classification, well, an assay type information in ChEMBL. So we have binding assays, ADME assays, tox assays, functional assays. Yeah, and functional assays, I think that's it. So if we have it as a filter, yeah, here. So you can use, so if you go to the assays page, you can then, you know, filter, for example, on all the, what we've marked as tox assays in ChEMBL. So this won't be complete. For example, I know that a lot of the cytotox assays are still marked as functional because it's quite difficult to know what is somebody sort of checking the viability of a cell versus testing, you know, it's an anti-cancer agent, so you know you're trying to get it to kill the cells. So you know it's not complete. We've started trying to do that classification, so hopefully that helps. So do you mean putting data into ChEMBL? So admittedly we haven't done a PubChem release for quite a long time. The UniProt cross references we should do, I think, with every ChEMBL release. And I'm not sure what EMEP,
European
PNC. So, well, if the compound, well, all the links that we provide in ChEMBL are all linked to well, typical, that's just a, so all the links that we provide are Europe PMC links. So I think if you go here, yeah, all our links are to in Europe PMC. So those, you know, every time we do an update and it, you know, the data is from a specific paper, then that is the link that we will provide. Yeah.
I think there's just one last question.
Yes, I mean, this is obviously something that we're doing all the time.
So that's the species.
Yeah, so I mean, I think probably the best thing you can do is if you email ChEMBL Help, yeah, then we can, and you know, explain a bit more what you're wanting. We can probably send you a reply and, you know, try and help you more specifically as to the best way of identifying the data that you're interested in. Because yes, some of this data is quite complicated and we don't have enough people to curate everything to the extent that we want to all the time. But, you know, seriously, do just email ChEMBL Help and we'll see what we can do.
So we'll leave the questions there for today, but as Anne says, if you do have any questions, check out the ChEMBL Help Desk and they'll get back to you and help you out. As mentioned at the beginning, there's going to be a little feedback survey, very short survey, that pops up right at the end as this webinar finishes. If you are able to tell us how you found today's webinar, that would be great for us. And lastly, as we mentioned, this has been recorded and we'll also make the slides along with this recording available on Train Online, and we'll send out an email tomorrow to let you know when that's been made live. Okay.
And I mean, I didn't go through the slides, but there are a lot of screenshots that tell you how to navigate around the interface that I've also provided. Yeah, and that'll be part of the slide set.
Yeah, that will be available for download along with the video. Okay, well, thank you, Anne, for joining us today and giving us this useful presentation. And thank you all for logging in and joining us and asking your questions too. Great. I hope to see you at another webinar in the future.